Hey y'all, hope you're doing good. We are moving on. Hopefully, you feel confident with chapter 17 with the um, endocrine system. But we are moving on into chapter 18, which deals with blood. Now, we mentioned last time that the hormones, in order to function properly, they need to use the bloodstream, right? And so, whenever we look at that, that leads us into this chapter 18, dealing with the bloodstream. And so we are going to start to talk about all those components, all those different things that the hormone might encounter while it is in the bloodstream. So blood, first off, let's talk about cardiovascular system, because that's where we really need to start this thing, is at the cardiovascular system. So first off, the next three chapters are going to cover the CVS, or the cardiovascular system. This first chapter is going to be dealing with the blood, and this is the transport fluid. Again, this is a type of fluid connective tissue, if you've forgotten. So we'll talk about that here in just a second. And then we will talk about, and again, it's going to be this chapter. And oh, I kind of messed that up, didn't I? I just realized that. I put the wrong chapters. Chapter 18 is blood, chapter 19 is heart, and chapter 20 is vessels. So sorry about that. I screwed that up. But either way, our next three chapters are on the cardiovascular system. Blood, along with endocrine, is on our first lecture test. Heart with blood vessels is on our second lecture test. So 1.5 of our test is dealing with CVS, right? So pretty important. So we've got the blood, that's the transport fluid, the heart, that's the pump, and then the vessels, which contain the blood and just send it around the body. Now, again, whenever we're looking at blood, this is a type of fluid connective tissue. If you've forgotten connective tissues, we've got CT proper, we've got supportive, and then we've got fluid. If you remember, the fluid connective tissues were the transport mediums of the body. And don't forget that connective tissue has three main components: cells, fibers, and ground substance or matrix. Now, in the bloodstream, we've got more than just cells. We have platelets. Platelets are not living, they're not considered cells. So, as a result, we need to call what these cells are in the bloodstream something different. We're going to call them formed elements. So now we're going to talk about red blood cells and white blood cells and platelets as being our formed elements in the blood. Okay. Now, we don't have fibers in blood. You've probably never heard of collagen or elastic fibers in the bloodstream. And if you have, you're in trouble, right? So instead of fibers, we have something called plasma proteins. If you remember, fibers are just types of proteins. So here, fibers usually give physical strength. Now, these plasma proteins are going to give us other physiological functions. Okay, so we'll talk about that. Each group of these, we've got three groups, are going to kind of do a little something different. Now, again, we've got cells and we've got plasma proteins, but surrounding that, we need the ground substance, and that's going to be the fluid, which is plasma. Now, plasma is primarily water. A little bit more than half of your blood is plasma, and about 95% of that plasma is water. So somewhere around half of your blood is actually just water. Now, the plasma is where we dissolve things for transport, or this is also where we store our water. We store water inside of our cells in the cytosol, but for hydration, we're usually talking about exchanging water in and out of the bloodstream to create our hydration. If you're dehydrated, you don't have as much water as you need to in the bloodstream. And if you are overhydrated, you've got too much. And so, where do you store water? Unless you're a camel, you don't store it on the hump on your back, right? So we store it in our bloodstream. A lot of people don't realize that. Now, blood. So here's the components, right? We've got formed elements. Instead of fibers, we got plasma proteins, and the matrix or ground substance is plasma, and that is the fluid. Now, we are continuously regenerating our blood. We have approximately 25 trillion red blood cells in our body. So we're constantly regenerating the blood cells. We're constantly adding and taking away from the fluid composition of the plasma. And so we're constantly changing and regenerating this blood. If we lose too much, it's obviously fatal. It's important for transportation of gases, nutrients, waste, hormones, good stuff, bad stuff. And as I mentioned, because it's transporting different things, we've got different amounts of fluid, we've got different amounts of other things that are happening. It's an ever-changing composition. It's a very dynamic fluid. Now, I mentioned just a second ago these blood vessels, so I do want us to make sure that we're familiar with the basics of the vessels, even though this test is not really testing on that. There may still be a question. I want you to be familiar with it. Three basic vessels, and I hate that they put these out of order. Arteries go away from the heart. They take blood away. They lead towards capillaries. Arteries get smaller and smaller and become capillaries. Capillaries are our exchange vessels. Anytime you see capillary, there's your word connection, exchange, right? So here's where we exchange good stuff out of the bloodstream into the tissue, and we're going to take that tissue's bad stuff and put it into the bloodstream, right? Then we're going to send it back to the heart through a vein. So veins are vessels that go back to the heart and take that used blood that we exchanged at the capillary back so we can start to refresh it, right? Start to take the waste away and get more nutrients and more gases. Now, as I mentioned, the blood components. There is an image in our textbook that is a very helpful image, and I'm gonna zap over there just real quick, and then I'll come back. Here's this image. Okay, this image is, I think it's still figure 18.1. Here it's showing us if we centrifuge blood. Here is a sample, but it's separating blood and it's showing us the components right here. This information is our entire chapter in a nutshell. Okay, so when in doubt, reference this, right? So there's three components really. There's these, or two components. There's plasma, and then there's formed elements, right? Most of the formed elements are going to be erythrocytes, red blood cells. So 99% are going to be reds out of all of the formed elements. Only a small amount are going to be, and we can see the numbers here, millions. Here we've got hundreds of thousands, and here we've got 10,000, right? And so there's going to be a lot less whites, and there's going to be kind of a medium amount of platelets. So these are going to equal the formed elements. Here is plasma. It is mainly water, but it also has these three main groups of proteins: albumins, globulins, and fibrinogens, right? And then we put all the stuff that we're dissolving, all the things that we're floating, or maybe some of our enzymes, regulatory proteins. We're going to put those into that plasma as well. Okay, so when in doubt, use this image. Just that, this kind of three boxes right here helps to separate everything and keep it kind of straight in our head. So here it is again, kind of just spelled out for us. Whenever we look at whole blood, first off, I want you to know that whole blood is plasma plus formed elements. So those are the two main components, formed elements plus the plasma. Again, formed elements, instead of cells, we have formed elements in the blood. Erythrocytes are the red blood cells, leukocytes are the white blood cells, and platelets. And again, platelets are actually cell fragments, and that's the reason that we can't just call them blood cells, okay? The reds are involved with carrying gases, especially oxygen. The whites, I'm sure you know, are involved with defense against pathogens, our body's immune system. And platelets, I'm sure you're familiar, involved with clotting blood. Okay. Now again, plasma is that fluid portion, and it mainly contains the fluid plus those proteins instead of fibers. We don't need strength in blood, we need more function. So we're going to have these plasma proteins, and then whatever we've dissolved, whatever we're transporting, is also in that plasma. Now, let's look at some functions real quick of the blood. So functions of blood, our first one is the obvious one, transportation. That's our primary function. We're transporting gases, right? We're transporting good stuff, nutrients, hormones. We're transporting bad stuff, waste. We're even transporting heat. Your blood is a radiator fluid, right? It takes heat from where it's made in the muscles, and it transports it to the skin so it can be radiated out. So our blood is our delivery system for the body. Blood is also involved with regulation. As I just mentioned, regulating body temp. It absorbs heat from cells that make it, from your muscles, or maybe those cells that are experiencing that calorogenic effect that we talked about last class dealing with endocrine. And so it absorbs heat, and then it takes that heat and it releases that heat at the body's surface. Now, we're going to find out here in a second when we look at characteristics of blood. If blood is carrying heat, it's going to be hotter than the rest of your body. So 98.6 is your basic body temp that you should be. Blood is going to be about 100.5, 100.2. So it's going to be almost 2 degrees warmer because it is acting as a radiator fluid. Your blood is involved with regulating pH. It's involved with buffers. So buffers, don't forget, have the ability to bind and release hydrogens. It's hydrogens that create a bad pH, create that acidity or the basicness, the alkalinity. And so as a result, if we have these buffers that can bind and release these hydrogens so they're not in the blood floating freely, then they won't affect pH. The blood, if we can balance the blood, then the cells will balance too. And so balancing the body pH is pretty easy if we can balance what's happening in the blood overall. As I mentioned before, it's also going to be involved with fluid balance, hydration overall, right? So we're going to add water by drinking it, getting it through the intestines. We're going to lose it by sweating or by urinating. And in between, we're going to contain it in the blood. So our blood is our storage for water. It's our water storage tank. And so as a result, it's going to be involved with fluid balance. We're going to see other things, some proteins, some ions, sodium, things like that are going to be involved with this balance as well. The last function is involved with protection. Here we can mention our white blood cells involved with immunity. Some of our plasma proteins, specifically our antibodies. Antibodies are types of plasma proteins. They're called immunoglobulins. And so these antibodies are also important to help bind to any foreign antigen and target that bad guy, target that cell and try to kill it. But also protection from blood loss, from fluid loss overall. That's what platelets are doing, and some of the fibrinogens, another group of plasma protein, are important in clotting. Now, let's move on from functions to characteristics. So now let's talk about what are some of the things that makes blood, makes blood different from everything else. Well, one thing is its color, right? The color is dependent on that hemoglobin, and we're going to talk about that here in just a second. So hemoglobin is what binds that oxygen. If it's got oxygen bound to it, it's a brighter red. If it doesn't have as much oxygen bound to it, it's a darker red. It's more of a duller red, right? Now, the reason why it appears blue in our veins is not because, well, this is like some genius. Due to the way blue light is reflected at the eye. No crap, that's how all light works, right? It's reflected back at the eye. Don't be a moron. Dark red blood in your vein. It's the vessel wall of the vein that gives it that blue appearance. The dark red of the blood plus the vessel wall of that vein now makes it blue, right? And then because our eyes always reflect, they interpret and reflected light, then that blue light kind of enters our eyes and we see that. Anytime we see blood, in general, unless we're in a vacuum, it's going to be bright red. If you cut yourself, well, then it's exposed to the oxygen in the atmosphere. So now your blood's automatically going to be bright red. If you are a blue-blooded American, right, you've heard that term, blue-blooded, well, that means you're actually from outer space. You're an alien, right? Because we don't have blue blood. We got red blood. So blue is simply just kind of a little trick of the eye, okay? Now, some more characteristics. How much? Volume. First off, I want you to realize this is the only thing basically that is not due to testosterone. That's not due to male-female differences. This is due to size. So go ahead and give yourself that note. Volume, you have more blood if you're bigger, less blood if you're smaller. That's all that it is. Okay, so males tend to have more than females because overall males tend to be bigger than females. But if you have two people, a male and a female, that are the exact same size, they're going to have the exact same amount of blood in general. Most people have a certain percentage of their body as blood. And so if you're bigger, you have more blood. It's somewhere around 5 to 6 liters or 5 to 6 quarts. Those are very close measurements. So 5 to 6 liters or 5 to 6 quarts. Usually males are going to be closer towards the 6 and females a little bit closer down towards the 5, that kind of thing. Now again, this is all due to simply size. Thickness, viscosity, another characteristic. Here, 4 to 5 times thicker than water. You've heard this before. Blood is thicker than water, but this is due to the dissolved substances. We've got other things dissolved in there. We've got cells, we've got platelets, we've got dissolved stuff that we're carrying, good and bad. So it's going to be thicker, okay? Plasma concentration. This is going to be also referred to as osmotic concentration. Okay, so we will talk about this as osmotic concentration more than anything else. Anytime you see the term plasma concentration or osmotic concentration right now, what it has to do is movement of water. And whenever we're talking about movement of water, what we're usually talking about is at a capillary. Okay, moving water in and out at a capillary. And that's what it says here, determines whether fluids move into or out of plasma by osmosis. So there's this characteristic that's referred to as osmotic concentration or osmotic pressure or plasma concentration. This is due to how much solutes are in the plasma. More solutes equals less water, right? We talked about this with hypertonic and hypotonic, but now we're going to use that and we're going to move water between the bloodstream and between our tissues. And so this is very important for that exchange at capillaries. Now, as I mentioned before, blood is carrying heat. It's acting as a radiator fluid, so it is warmer. It says 1 degree here. Most textbooks say almost 2 degrees. So 1 to 2 degrees higher. Usually it's 1 degree Celsius or so. So 1 degree or 2 degrees higher compared to your body temp because it is carrying that heat from where it's made to the skin where it can be radiated out. The pH. The pH is exactly normal for our body. And if you don't know that, you should know that now. Blood pH is between 7.35 and 7.45. This is simply our body's natural balanced pH. Even though it says that it's slightly alkaline, that doesn't matter. Our body's balanced pH, where our proteins like to work better, is in this range, somewhere around 7.4, and that's where our blood pH is right in that sweet spot. Okay, now let's move on. Let's talk a little bit about these getting blood, breaking it apart, and then we'll talk about its components. Okay, so first off, whenever we look at this, there are 3 ways that we can collect blood. And if you look at this and pay attention, you're going to notice that they're really collecting from 3 different areas: venipuncture from a vein, peripheral capillaries from your capillaries, and arterial stick or an arterial puncture from an artery, right? So it depends on what we need that blood for as to how we're going to collect it. Right. So if we just need to run a quick test and test some numbers, well, we may be able to do a capillary prick, right? But otherwise, we may need to take a little vial from venipuncture to get some information. What does an artery have that a vein doesn't, right? So we'll talk about that. Let's go ahead and let's talk about this though, and then we'll lead to that. Venipuncture. Venipuncture is the most common. This is where we puncture into a vein. Veins, especially the one in the bend of your elbow, the median cubital vein. It's very superficial, so it's easy to locate. Now, the reason that venipuncture is the most common method is, number 1, a vein wall is thinner. So there's less smooth muscle in it, so it's easier to puncture. So we can puncture the vein wall easier. Also, veins have lower blood pressure. So that means that instead of having a big spray of arterial blood coming out, the venous blood doesn't have as much pressure. It's not going to spray out, and therefore the wound is going to heal up faster, right? So we can tap a vein and get most information from the blood that we need, but there's certain things that we can't. For example, if we wanted to puncture an artery, what does an artery have that a vein doesn't? We haven't learned it yet, but usually an artery has oxygen. And so if we're looking to test for gas exchange, efficiency of gas exchange, then we're going to do an arterial puncture, an arterial stick instead. So the radial artery at the wrist or maybe the brachial artery near the elbow, we're going to puncture into that and we're going to draw some blood to test to see how much oxygen is in there. Is someone exchanging gases efficiently at the lungs or are they not, right? If we put them in an environment and can measure how much gases they should have in their blood, and then we measure their blood and they don't have that amount of gas, well, there's an issue, right? So that can help us diagnose a problem. Last thing is peripheral capillaries. If we do just a little finger prick, right, a heel prick or a toe prick, that kind of thing, okay? So these are sampling from just peripheral capillaries in your skin. And what this does is we can take a little sample to test for some very basic kind of things. You can get blood oxygen. You can do that with other things now too. You can test your glucose levels. So we could use it for testing, but many times we're using it to get a drop of blood also to create a blood smear. And a blood smear, you need to know, is simply just a slide of blood. A blood smear is simply just a way to look at blood physically to see maybe we can see sickling of cells and identify sickle cell anemia. Maybe we could see that your white blood cell count is extremely high from there, so we could take even some white blood cell count kind of information from a blood smear. So we're going to use a blood smear anytime we need to see the blood, like underneath a microscope. All right. Now, usually we're going to venipuncture, right? If we venipuncture, we take a good sample, then we're going to centrifuge that blood out many times. And whenever we centrifuge that whole blood, we're going to get its two main components. Remember, whole blood is plasma plus formed elements. So we're going to get those two main components spun out. We're going to spin out based on weight, right? So we're going to base this on weight. What weighs more, a solid or a liquid? Well, solids are more densely compacted together, so they're going to weigh more. So the solids are going to be spun farthest away. And what ends up happening is, let's go to this picture. When we put this tube in, it ends up kind of spinning it so that the bottom of the tube is farthest away. So the heaviest things, the solids, are going to pack down to the bottom, and the lightest things, the fluid, is going to pack towards the top. Okay? Now, we're going to get three layers. We're going to get an erythrocyte layer. This is the lowest layer. This is the bottom layer. And these are just your red blood cells. This should be less than half, a little bit less than half of the sample. Now, this depends on your hematocrit. And we're going to talk about that here in a minute. But this is how you measure hematocrit. This is a method of measuring hematocrit. And so this can change. This can go anywhere from 40 to 50, somewhere around there, okay? But if you are doping, if you're cheating in the Tour de France like Lance Armstrong, they're going to take a sample. If you've got more than 50, then you are suspect. If you've got 55 to 60, you're cheating, and they know it, right? And so they can test you very quickly with a simple sample, centrifuge it for a few minutes, and bam, we can see if you're cheating. Now, the middle layer is really hard to see because it's less than 1% of the sample. This middle layer is called the buffy coat, and the buffy coat is kind of whitish gray. Okay, this is where the other two formed elements hang out. The whites and the platelets are in the buffy coat. So many times it's really hard to see that buffy coat. It's almost kind of just like this extra little layer that kind of floats there, and you don't really see it that easily unless you kind of agitate it a little bit. And then on top, we're going to have more of the straw-colored liquid. And that straw-colored liquid is the plasma. Again, this is where we're going to dissolve everything and kind of float it through the body, transport it through the body. Again, here's our process. Here's a venipuncture, and then here we're going to spin it centrifuge, and we're going to get its components, right? So we get some reds, which is a little bit less than half. The whites and the platelets are less than 1%, and the majority of it should be that plasma. Okay, again, when we look at plasma, it's got water and it's got those plasma proteins, and then it's going to have whatever we're carrying. Okay, here again, looking at that centrifuged blood, I just mentioned this term hematocrit. Hematocrit is percent of red blood cells. Some people call it an RBC count. It's not a real count, it's really a percentage. What percent reds versus everything else in the blood? Now, 99.9 of your formed elements are reds. So there's less than 1% of whites and platelets. And so usually whenever we're measuring this, we can spin it out. All the formed elements is going to equal your hematocrit, but it's really kind of giving us a percentage of erythrocytes. Now, here is where all the differences start to become due to testosterone availability. If you have more testosterone, you're going to have more red blood cells. So males are going to have a higher hematocrit. And this is what I was saying, that erythrocyte percentage that was on that image, it changes based on your hydration, based on how much oxygen you've been breathing, and if you need more red blood cells. So if you've been at high altitude, for example. And so males tend to have higher, up to 56%. Females not as much because they don't have that testosterone, right? They don't have testosterone. And again, as we mentioned last chapter, testosterone triggers EPO, and EPO triggers that red blood cell production. So that's going to increase it, right? Now, if you're a male and you're over 50% and you're in an athletic competition, that's kind of a red flag. So like I said, that's one of the ways that you can get busted trying to blood dope, trying to cheat, trying to alter your RBC count, okay? Again, that blood smear, very basic. I'm not going to go through these details because this is not going to be on the test. Just understand that we're making a slide of blood, and what we can do is we can get a good visual of it, right? So we can look at this under a microscope, and we can see all of these reds. We can see some whites. We can even kind of see a few platelet chunks kind of here and there, all right? Now, let's move on and let's talk about plasma. If you haven't noticed, this chapter is going to move a little faster than that last chapter. There was a lot to bog us down in that last one. This one's got a few topics that bog us down, but overall, we're going to move quicker through this because it's a little bit more basic information. Blood plasma, again, it's primarily water. 90 to 95% of the plasma is going to be water. And the other percent is mainly those plasma proteins. And again, these are what we had instead of fibers. Remember, this is a connective tissue. And so instead of fibers, because we don't need strength, we need other functions. So here is that plasma protein. And then we have the other stuff that we're carrying, dissolved molecules and ions. Plasma, blood, is the primary ECF, extracellular fluid, right? So this is the main fluid that's outside of your cells. It's similar to the fluid that's in your tissues, interstitial fluid, but this is going to have more plasma protein. So it's going to have a higher protein concentration compared to what's in the blood. This is going to encourage exchange. We're going to see that. This is going to set up, now we've got a hypertonic solution. Plasma becomes hypertonic. That means it's got low water, so water wants to come back into the plasma. We'll talk about that when we get to blood vessels. We'll talk about how that relates to exchange. Now again, blood's a colloid because anything that's a colloid is a protein mixture. It's just a fluid with protein in it. And these plasma proteins turn it into a colloid. And so that's just a term, that term colloid. Don't let it get you down or anything. But we've got three flavors: albumins, globulins, and fibrinogens. These are the main ones. Now we've got some other guys, but these are the ones that we're going to consider. Now you should be familiar that most of these are made in the liver. There's only one group, one of the globulins are immunoglobulins. And these guys are made by B cells, by some of our white blood cells. So all of these plasma proteins made by the liver except for our antibodies, one of the globulins, and those are made by our whites. Now, if you have liver damage, liver failure, then these are going to be affected. You're going to have problems clotting. You're going to have problems exchanging at capillaries. Okay, so definitely some of those side effects from liver issues. Albumins, this is the most abundant. You don't have to know 58%. Okay, I don't really like these kind of numbers for my class. I want you to know just kind of in general. So albumins are the majority of the plasma proteins, the most abundant. What I want you to be familiar with, they are involved with transport. I don't want you to worry about that as much. What I want you to focus on is osmotic pressure. These guys exert the greatest osmotic pressure in the blood. Now, again, anytime we see osmotic pressure, that has to do with exchange at capillaries. Helps to maintain the volume and the pressure, but it also helps to maintain exchanging at capillaries. So I want you to know that these albumins help set up exchange at capillaries. They help with osmotic pressure. Now, secondarily, they can be used as transporters, but I want to add transport to something else. So we want to focus albumins with osmotic pressure. Okay, and these are the majority. Now again, osmotic pressure, this is the same thing as tonicity. So this is setting up a pull that brings fluid back and waste back out of the tissues and into the capillaries. And that's really the key. And we'll talk about that when we get to blood vessels. So don't try to push that topic too much yet. Osmotic pressure, I'm not really harping on it right now. We're just kind of getting some basics. All right. The second group are globulins. And the globulins, we've got two subgroups. We've got the transport globulins and we've got immunoglobulins. Now, again, these are the second most abundant. And then our last one, our fibrinogens, are going to be the least abundant. But overall, we've got one group called transport globulins. These are the ones that can bind hormones and act like carriers. So that's the way to think about it. These are the carrier proteins in the blood, these transport globulins. And so they can bind substances and transport them inside the bloodstream for us, right? So these are little carriers, little ferries in our blood. This last globulin is an immunoglobulin. Sometimes these are abbreviated IG, big I, little g. These are also referred to as antibodies. This is mainly what I'm going to call them are antibodies. So antibodies, very important for our body's defense. If you can't make antibodies, you will die. Okay, HIV inhibits the body from creating antibodies overall, and therefore you're ending up going to die. So not a good thing. So these immunoglobulins and these antibodies, super duper important. Again, this is the only one not made at the liver. All the rest of these that we're talking about right now, these plasma proteins, are made at the liver. Fibrinogens are the last. So fibrinogens, a very minimal, minor percent. You don't have to know 4, just know that it's the lowest in percentage in the blood. And so here, fibrinogens are involved with clotting. Without fibrinogens, you don't make fibrin. And fibrin are kind of like some of the logs in a beaver dam. So it's hard to make a dam and hold water back if you don't have logs. And so here, this fibrin is kind of a very important component of those blood clots. Now, whenever we whenever we take these clotting proteins out of the plasma, fibrinogens can start to clot without you wanting them to. And so if you remove the fibrinogens from blood, we get something called serum. So whole blood minus fibrinogens is going to be our serum. Some of that blood, once you take it out of the body, it could just start to clot. That's not a good thing. Now, there's also some other regulatory proteins. We're not going to worry too much about this, but enzymes and hormones and things like that. There's really 3 main plasma proteins. This is something extra. This is part of what is being carried in the plasma. So don't worry about these regulatory proteins as much. Now let's look at the formed elements, and that's really kind of what the rest of the chapter is focused on, is the formed elements. So whenever we look at this, there are 3 formed elements. There's the reds, the whites, and the platelets. Again, a little bit less than half. Reds involved with gas transport, whites involved with defense, and platelets involved with clotting. So let's first jump in and talk about overall the process, hemopoiesis. Hemopoiesis is the overall process of formed element production. So making the formed elements is called hemopoiesis. Now you need to know this takes place in our red bone marrow. Okay, so I've heard it called hematopoiesis as well, but this is taking place in the red bone marrow. And what this is when the stem cells are spitting out the cells that are needed. Well, we've got 2 kind of layers of stem cells. We've got the main stem cell, and then we've got 2 other groups of stem cells that it makes, and those 2 groups of stem cells make the rest of the blood cells. There's a great chart. Unfortunately, I'm going to zoom over here, and you're going to see half of my chart's kind of missing, and so it really screws all this up. It makes it so it's not as understandable. But look at this chart in your textbook. Here's the main stem cell. Here's the other 2, and then here's what they make down here. Okay, and you can see this a lot better in your textbook. Here's what I want you to know. The main stem cell is called a hemocytoblast, blood cell builder, right? That's what blast means, is to build. And so hemocytoblast, this is the main stem cell. Now what it's going to do is it splits into 2 other stem cells. It creates a myeloid stem cell and a lymphoid stem cell. Okay, so the myeloid stem cells and the lymphoid stem cells. Here's the way to memorize this very easily. Lymphoid stem cells only make lymphocytes. We haven't got there yet, but lymphocytes are just one of the 5 types of white blood cells. So the lymphoid stem cell only makes lymphocytes. The myeloid stem cell, on the other hand, is going to make all of the other formed elements, the reds, the platelets, and also the rest of the whites. Now, hemopoiesis is the overall process, but whenever we break it down, we can also talk about the specifics of each one of these formed elements. Erythropoiesis is the specific process of creating red blood cells, erythrocyte production. So if you haven't figured out yet, the suffix poiesis means cell production. Leukopoiesis, white cell production. So this is the process of making white blood cells. And then the last one is called, don't worry about that right now, thrombopoiesis. Thrombopoiesis is the process of making platelets. I don't care if you know megakaryoblast and megakaryocyte. Don't worry about those. Just know the terms erythropoiesis, lymphopoiesis, and thrombopoiesis as far as what they create, which specific formed elements that they're making. Again, here's that chart. Take a look in the textbook. Hemocytoblast makes the myeloid, makes the lymphoid. In our textbook, if you can see this, if you follow it down, here's our lymphoids. That's it. Here's our lymphocytes. All the rest of these whites and our platelets and our reds are made from the myeloid stem cell. So again, hemocytoblast makes a myeloid and a lymphoid stem cell. Lymphoid only makes lymphocytes. Myeloid makes all of the rest. And here's just kind of that process, right? We got the bone marrow, we got these stem cells, and so these guys are going to be spitting their here this is showing platelet formation, spitting their formed elements into the blood. Now let's turn our attentions towards the red blood cells, and then we'll talk quickly about whites. We do have a full chapter dealing with immunity, and so we're not going to get into too crazy much details about these whites, and then we'll look very quickly at platelets. So first, the majority, like I said, of what's left, which is a little bit more than half of this chapter, is going to be with the red blood cells. So red blood cells are erythrocytes. These guys are small and they're flexible. They have to be able to bend in order to get through some of the smallest blood vessels in our body. Again, these are our RBCs. These are highly specialized cells. These are, besides the sperm and the egg, these, in my opinion, are the next most highly specialized cells in our body. Okay, the sperm and the egg, extremely highly specialized just for reproduction. These guys are one of the most highly specialized of our body cells, right? Instead of cells, these are dealing with body cells. These guys don't have a nucleus. Red blood cells don't have a nucleus, and they don't have any organelles, right? All that they are is a plasma membrane with some cytosol, and in the cytosol is a whole bunch of hemoglobin, okay? These guys are shaped like thick crust pizza, skinny in the middle and fat around the edges, biconcave. That's what that means. Now, what's happening here? The whole job of this RBC is to transport oxygen. So we don't want a nucleus or a mitochondria or any other organelle that might consume that oxygen. If we're trying to carry something, and we want to keep it available for something else, then we don't want to use it up, right? And so we want to get rid of these organelles so that we don't consume the oxygen that we're carrying. And it's that hemoglobin molecule that carries that oxygen. Now that shape is important because it gives us, let's take a look, instead of being round, it gives us more surface to volume ratio. And so as a result, we can exchange. Here you kind of see that biconcave, fat around the edges, skinny in the middle. What that does is it gives us more surface area to exchange oxygens with that hemoglobin in and out of that red blood cell, and it happens extremely quickly. And so that surface area due to that biconcavity is a good thing. Now, these guys mainly transport oxygen, but they can transport CO2, and they do. They transport about a quarter of the CO2, but they transport 90% or so of the oxygen. They can form these stacks. I always like to think about checkers. If you just stack a whole bunch of checkers up, we can do that with erythrocytes. We can do that with these reds. We can stack them up so they can go through some of those smaller blood vessels, and we call that a roulet. Okay, so a roulet is just a stack of these red blood cells. Don't worry about spectrin, okay, and the flexibility aspects there. Here we can kind of see a roulet starting to form and going through that blood vessel right there. Kind of interesting. Now again, the key here to these reds is hemoglobin. Hemoglobin is a protein molecule, and it is a red pigmented protein. Most proteins have a color. That's where we get most of the colors that we see is due to protein pigmentation. And so here, this one's red. Again, if it's carrying oxygen, we call it oxygenated. If it's not, it's deoxygenated. I don't know why these words are bold-faced. That's something you should already kind of understand, right? Oxyhemoglobin versus deoxy. With oxygen, without oxygen. It's not that complicated, right? And so this determines our color as well, a bright red or a dark red, right? I'm not too concerned about carbaminohemoglobin because you're always carrying carbon dioxide on there. So it's always carbaminohemoglobin, and it's either oxy or deoxyhemoglobin. So don't worry about that last one. Let's just focus on oxy and deoxy. Now again, this is a red pigment. Hemoglobin contains 4 subunits, 4 globins, okay, 4 subunits, 2 alphas and 2 betas. I don't care if you know that. I just want you to know it contains 4. It contains 4 subunits, and each subunit has 1 heme group in the middle. The heme group is where the oxygen binds, and you need to know that one of the key features of this heme is that it contains iron in the center. The oxygen binds to the iron in the center of that heme group. Now, what happens when you bind oxygen to iron? Iron oxide is rust. What color is our blood? it's kind of rusty, right? And so this is really what's going to help give it that coloration. Binding oxygen to iron produces that deep red coloration. Okay, so each hemoglobin, though, in the long run, has the capacity to carry 4 oxygens. Okay, now inside of each red blood cell, there are millions of hemoglobins. And then again, we've got 25 trillion red blood cells. So in the long run, we have got bukus of hemoglobin to carry bukus of oxygen. That's kind of the point. Okay, so again, oxygen can bind and unbind to hemoglobin very quickly. Now, the reason it can do that is because the binding between the oxygen and the hemoglobin is very weak. So this allows it to associate and disassociate quickly, to attach and detach very quickly. Okay, when we go through the lungs, we can quickly pick up the oxygen because it's only got a split second, right? It's got less than 2 seconds of time in the lungs. And then we can allow that to detach when we get closer to the body tissues because they want that oxygen to stay alive. Same kind of thing with carbon dioxide. It's fairly weak too. We can bind CO2 and we bind it from the tissues and we take it to the lungs to be gotten rid of. Here's these 4 subunits. Each one has a little planet Saturn. That's what it looks like in this image. But in the center of that is that red iron ion, and that's where that oxygen binds, right? And so it binds there and gives it that red coloration. Now, again, don't forget that erythropoiesis is the process of red blood cell production, and the hormone that triggers that is EPO, erythropoietin, right? So erythropoietin is what triggers RBC production, which is erythropoiesis. Now, the kidneys are what make that EPO. Here it talks about the liver makes some of it too, but again, we're mainly talking kidneys, all right? Now, I'm not really concerned that you know this, but what would stimulate more red blood cell production? What would stimulate more EPO? Decreased blood oxygen. So if you go higher in altitude, if you exercise too much, if you're losing blood, right? It's detected by these chemoreceptors in the kidneys. EPO is released, goes to the bone marrow, and it triggers the myeloid stem cells to produce more of these RBCs. Okay. As I mentioned, testosterone has an effect. So testosterone it's kind of a permissive, kind of a synergistic effect with this EPO. And again, that EPO can be triggered easily by altitude. Higher altitude has lower oxygen available, and as a result, the body wants to release that EPO so that we can gain more red blood cells and carry more oxygen. This is why athletes train at altitude. They're trying to increase their hematocrit legally, right? Legally. The problem is if you increase it too much, or you increase it at all, but especially too much, you're increasing the thickness. And the problem now is you're forcing the heart to work harder, and that can increase complications in the cardiovascular system. So definitely not a good thing. That's what blood doping is. So blood doping is not the same thing as using steroids. It's not the same thing as trying to build big muscle. Blood doping is simply trying to carry more oxygen. Now, the easy way to do that is by having more red blood cells. So there's 2 ways to do that. The first is called self-donation. You can actually take blood out a month before your competition. Your body will regenerate that blood, and then right before that competition, you add that extra liter of blood back into your body. So now you've got an extra liter's worth of blood cells, red blood cells in your blood, and that's going to carry more oxygen compared to your competitors. But again, now you've just increased the viscosity and you've increased the blood volume. So now you're starting to do damage on the heart. So we're going to see Lance probably dying of a heart attack, because he's done so much damage to his heart. Even though he's an athlete, he's done serious damage by using these drugs. The second method is by using EPO, by simply just injecting EPO, and then that way it triggers that increased blood count. Now, as I mentioned, we're going to increase the viscosity that forces the heart to work harder, and now we're starting to cause permanent cardiovascular damage. It wasn't just the chance that he might have a heart attack while he was doping. Now he has caused permanent damage to his heart. And so that's the reason I said we're more than likely going to see Lance die of a heart attack eventually. And that's a tough thought, because I was one of those back in the day whenever he was doing this and he was claiming he was clean. I wanted to believe it. He was a superstar. He was a superhero on that bike in the Alps and in the Pyrenees. And so, I wanted to support him personally for a long time until he really kind of admitted it, and it just became real evident and obvious. And so, but in the long run, he's done what he's done, and he's going to have to pay for it, possibly, with heart damage. You're going to be banned from athletic competition. Right now it's 2 years, if I remember. At least in Tour de France, you're out for 2 years. And if you know anything about cycling, you don't have but maybe 3 years of your prime as a cyclist to possibly win a tour, win a stage, something like that. So you pretty much just ruined your entire professional career by doping. Not a good thing. Destruction. So after about 120 days or so, these guys, they can't repair themselves. They don't have any of the organelles to do it. So after about 120 days of being rolling through the system and rolling through the blood vessels, they're going to get beat up and their membranes are going to start to rupture. So once they enter the spleen or the liver, they're going to be phagocytized. They're going to be eaten and they're going to be recycled. So we're going to recycle most of these parts. I'm not going to get into details, but we're going to store that iron in the form of ferritin, ferrous, Fe, iron, right? So we're going to store that especially in the liver, and then we're going to recycle as much of the other stuff as possible. I'm not really going to go into details about that, though. Let's mention very quickly anemia. Anemia is where you don't have enough oxygen carrying capacity in the body. So you can't carry enough oxygen. Decreased oxygen delivery to the body tissues. Something's going on and you don't have enough. Either you don't have enough red blood cells, those blood cells don't have enough hemoglobin, they're non-functioning, something's happened. Okay, so anemia is where you're not carrying enough oxygen. This doesn't mean that you don't have enough blood. Some people hear that, they think that they have low blood count or low red blood cell count. It could be low red blood cell count, but it could be other things. So it could be due to decreased formation, abnormal, heavy blood loss, genetic defects, some sort of deficiency. So anemia has many different forms. It can be treated with EPO, right? I want to mention sickle cell anemia. Sickle cell anemia is a unique form of anemia, and I want you to be familiar with it. If you take a look in your textbook and you find this anemia clinical box in there, then you can read a little bit more details. But sickle cell anemia is, I want you to be aware, it's a genetic disorder. It's a genetic mutation of hemoglobin. Okay, so hemoglobin is not properly formed. It's got a different amino acid substituted in there. And so what happens, though, is when the cells become deoxy, they tend to curl up. They tend to sickle. So they tend to look like half moons. They look like crescents. And so now the big problem is, if we've got a whole bunch of crescent moons floating in our bloodstream, they like to, barrel of monkeys is what I call it. They like to link together. You know how whenever you played that game Barrel of Monkeys, you used to try to pull one monkey out of that barrel to start the game and all of them were attached, because all those curls, all those hooks, all those sickles like to link together. So now with sickle cell anemia, the problem is these cells like to cluster together and now they form floating blood clots. Now we got a big problem. Okay, floating blood clot ends up getting lodged in a vessel, and downstream we have tissue death, right? We have problems. So definitely not a good thing. This could lead to a stroke, could lead to heart attack, could lead to amputation, can lead to problems all around, gangrene. So definitely not a good thing. Now, why is sickle cell anemia still around? Let's touch on that real quick. I want you to be aware that sickle cell anemia, it co-evolved with another disease. It co-evolved with malaria. So here's the deal. If you are a carrier of sickle cell anemia, then you are immune to malaria. And malaria is still one of the top 10 killers in Africa, for example. And so the African population especially has seen this as an advantage. If you are at a if you are a carrier, you don't have sickle cell anemia, but you could pass it on, then you are immune to malaria. So for a long time, malaria killed a lot of people. So if you were born with sickle cell, you may die of sickle cell. If you were born completely normal, well, then you may die of malaria. But if you are a carrier, if you've got one big and one little, whatever, S, right, then as a result, you are immune to sickle cell. You just got an advantage. So what happens is that this naturally selects for all of the carriers. So then a lot of the population, a lot of the true African population, for a long time now, things are starting to get better with malaria, and so we're starting to see this change a little bit. But that's the reason that sickle cell anemia is still prevalent in the African population and still even prevalent in African Americans. Even though these African Americans came from Africa, their genetics, once they reached America, they didn't breed with these same individuals. So they're starting to kind of breed this out a little bit of the African American population. And so in Africa, you're going to experience a lot more sickle cell disease than you will in America because we don't have to deal with that malaria, right? And so in Africa, it gave an advantage. In the US, it doesn't. And so as a result, and also there's a lot more genetics that are getting diluted in that process by breeding with many other people that haven't had to see this as an advantage. Okay, so sickle cell, really interesting kind of disorder. I like to talk about that, and it may hit home with some of you because you or someone you know may be dealing with sickle cell. And so I always like to talk about these things just to kind of give you a heads up and kind of relate and give you more information about it. Now, to start finishing up our talk about red blood cells, we're going to talk about blood type. And when we talk about blood type, what we're really talking about is what we're going to go into after that, immunity. Okay, blood type truly is the best example, the first example that we have of how our immune system works. So I want you to understand that. So give yourself that note, blood type, example of basic immunity. Okay, now here's the deal. On the surface of our cell, these little markers that identify that cell to the rest of our body, especially to our immune system. These markers are called antigens. So jot that term down. An antigen, marker on cell surface, identifies cell to immune system. Now here's the deal. These antigens, if they are normal, if they're the ones that are supposed to be there, immune system leaves it alone. So if you have self antigens, a self antigen is left alone. A normal antigen is left alone. But if you have a foreign antigen, then that foreign antigen identifies that cell as a bad guy. And now the immune system targets that foreign antigen and that cell and destroys it, kills it. So the key here, what is an antigen? Let me describe this real quick. An antigen is simply just a leftover scrap of protein that the cell makes. So if an antigen is just a little scrap of protein that the cell has made, what this antigen truly is it's an indicator as to if that cell is functioning properly. Is it making the right protein? Is it doing it at the right pace? If it's not making the right protein, then that means its DNA has been mutated. Its DNA has been changed. So truly what this antigen is an indication of is the quality of the DNA. Is the DNA normal? Is it functioning like it's supposed to? Or is it infected? Is it screwed up? Is it mutated? And now it's making something it shouldn't. Okay, so again, the normal antigens or self antigens are left alone, and the foreign antigens are targeted. Now, how do we target foreign antigens? Our immune system targets antigens with antibodies. And we mentioned this just a second ago. Antibodies. Here's the deal. Give yourself a note. If an antibody ever binds to an antigen, we're going to destroy that cell. So this is how we kill it. Binding an antibody to an antigen trigger self-destruct. That's the way you can think about it. And so if we have an antigen antibody complex, if we bind the antibody to the antigen, now we're having an immune response, and we're going to call in all these other cells that are going to destroy and kill that target cell. Okay? So blood type is based on surface antigens. So these identifying markers allow that blood cell to have its type and to be identified to the immune system. We've got two groups of blood types. We've got the ABO blood type, and then we've got the plus or minus, the RH blood type. Okay, so we're going to talk about both of those and kind of combine them. So you're familiar with, you may be A positive, right? So you've got the ABO group, and then you've got the plus or minus, the positive or negative. So it's really two different antigens. The first group, the ABO, is dealing with our first groups of antigens. There are two of these antigens. There is surface antigen A, and there's surface antigen B. Now, what they don't tell you here, they kind of try to describe it right here, but let's use some genetics words. Antigen A and antigen B are both co-dominant. These are both co-dominant. If you receive one or the other from your mama and your daddy, that's the way you think about it. So you got two of these letters that make up your blood type. It's either AA, AO, AB, or BB, BO, or OO. So those are the possible types of blood, right? If you've got an A anywhere, it's going to be expressed. And if you've got a B anywhere, it's going to be expressed. Okay? So if you are type A, then you only have surface antigen A, right? If you're type B, you only have surface antigen B. If you're type AB, you inherited A from your mama and B from your daddy, or opposite. You got A from one and B from the other. And so you contain both antigens. And if you're type O, you don't have either antigen. There's not a type O antigen. Type O means without antigens. Now here's the deal. If you have a certain antigen, your body is going to make an antibody to destroy the rest of those, right? The opposite. So if you are type A, the foreign antigen is type B. So if you are type A, I want you to give yourself a note. We can kind of see this coming over here. If you are type A blood, then you are going to have anti-B antibodies, because it's that type B that's foreign. And so you're going to target and destroy type B with these anti-B antibodies. So if you're type A, you have type A antigen and you have anti-B antibodies. If you're type B, you have the antigen B, but you also have anti-A antibodies. Okay? Because you're going to destroy A because it's foreign. If you're type AB, you've got both antigens, so you can't have either antibodies or you would destroy your own blood. So AB blood has neither antibodies. If you don't have any antibodies, that also means you can accept anybody's blood, right? So that makes type AB the universal recipient, okay? Type O, it has neither of these antibodies, so it's neither antigen, excuse me, so it's got to have both antibodies. And because it's got both antibodies, it's going to destroy any other type that it gets. So type O can only get type O, right? That's the reason when they're in emergency, they always need type O blood, because number one, type O can only take type O, but also type O doesn't have any antigens. So it can it can donate, sorry about that. It can donate its blood to anybody else because there's no antigens that would trigger a reaction. So because type O can donate to anybody else, they're also called the universal donor. And that's the reason type O is so important. Type O can only take type O, but type O blood can also be given to anybody else, okay? Especially O negative, right? And let's talk about that here in just a second with that other system. Now here is a visual, and this can help you kind of understand this. Surface antigen A are spheres, surface antigen B are cones. So the antibodies are going to match up to the opposite. So if you are type A blood, you're going to have anti-B antibodies, and they're going to match up with those cone-shaped structures, right? If they ever match up, they blow it up, they kill it. It's not necessarily blow it up, but we'll talk about that in other chapters. If you're type B, you've got surface antigen B, and you're trying to kill the foreigner, which is type A. So you're going to have anti-A antibodies. If you're AB, you can't have any antibodies or you'll kill yourself, right? So that means you can accept anybody's blood because you won't destroy anybody's. But type O doesn't have any antigens at all. It's got both antibodies, but because it doesn't have any antigens, it can give its blood to anybody, and nobody has antibodies to destroy it, right? And so that's the reason this is our universal donor, and there's our universal recipient. Now, what happens whenever we have a reaction? A reaction where we have an antibody attaching to an antigen, we're going to cause what's called agglutination. Agglutination is a clumping process, okay? So it's where we chop and we clump up these cells together, these bad guys, these foreigners together. And the problem is now they can start to block a blood vessel similar to that sickle cell. So not a good thing necessarily. And that's the reason that we don't want to have a bad blood transfusion. It can cause this incompatibility, and it can cause agglutination. It's going to cause hemolysis. We're going to rupture those cells, and then we're going to clump them together, and then it can block a blood vessel. So we've definitely got to make sure prior to donation that we do a little donor recipient test and make sure that they are compatible with each other. Okay, so here's that process, kind of agglutination. We're going to start to grab them together and clump them, but we are going to rip them apart and just destroy them as well, rupture them. Now, here's that second group of antigens, the second group of antigen D. We skipped antigen C. I don't know, they just hated C. They just kicked it out of the nest and said they never wanted it back. So I don't know what happened to antigen C, but we went from A and B, and then we went to D. So we are at surface antigen D, and surface antigen D is our Rh factor. So the Rh factor is simply just another antigen that must be paid attention to, or for a long time it really needed to be paid attention to for blood transfusions, especially for mamas, for pregnant mamas. Antigen D, if you have it, you are Rh positive. If you do not have it, you're Rh negative. Okay, so if you have it, you're Rh positive. If you don't have it, you're Rh negative. Now, here's the thing. If you're Rh negative, you don't naturally make antibodies. So when you're born, it doesn't matter if you're positive or you're negative, you don't have antibodies generally. Now, if an Rh negative person is exposed to Rh positive blood, so if I'm Rh positive and I get a bad blood transfusion and somebody gives me if I'm Rh negative, somebody gives me the wrong transfusion with some Rh positive blood, now I have been sensitized, I've been exposed, and now I'm going to start to make antibodies. Okay, so this, like I said, is not usually an issue. Let's take a look real quick. Here's our slide. These cubes are going to be our antigen D on the cell surface. Again, if you're positive, you shouldn't have any antibodies. But if you're negative, it seems like you should, according to our ABO system. But here, you don't make them unless you become exposed to that positive blood. Now, how would you become exposed? This has to do with mamas, and this used to be a major issue with mamas in pregnancies, especially the second pregnancy. Now, again, RH, we're going to do that plus or minus. So here, AB plus, A minus, so there's your blood types, okay? Now, here's back to that incompatibility with pregnancy. It was referred to as the hemolytic disease of the newborn, and what happened is, if mama is Rh negative, but her first baby is Rh positive, so during the first childbirth, she's Rh positive. Mama now becomes exposed, and now mama makes the antibodies. In a future pregnancy, if she has a baby that's Rh positive, these antibodies can cross the placenta and they may destroy the baby's blood cells. And then that would naturally abort that fetus, right? And so this is where it used to be a lot more important to pay attention. Nowadays, they just give pregnant Rh negative mamas a special immunoglobulin shot, a special antibody shot while she's pregnant, and everything works out just fine. So there's not an issue anymore. But this used to be a real issue with second babies that may be miscarried due to the first baby exposing mama and her starting to make her antibodies. So that's it for reds. And again, we kind of finished talking a little bit about immunity, and so that leads us into the whites. So let's move into the whites. I've got maybe 25 more slides or so left. And so I'm looking hopefully at maybe 30 minutes or so to finish this up. So white blood cells, leukocytes, right? These guys are involved with defense against pathogens. Don't forget pathos means disease, gen means to generate. So a pathogen is something that generates disease, right? So helps with defense. These guys, compared to reds, have their normal stuff. And they do not have hemoglobin, right? And these guys are more flexible and they're mobile. These guys are really cool. White blood cells, there are 5 groups, and you should go ahead and take a note about them real quick. There are neutrophils, there are eosinophils or eosinophils, there are basophils, there are lymphocytes, and then there are monocytes. So these are our 5 groups of white blood cells. Let's talk real quick about some of their characteristics. A couple of characteristics, and this is super unique, so make sure that you understand this process of diapedesis and emigration. Diapedesis means that they can squeeze between the cells in blood vessels. What that does, and I've said this before, the only place that we can exchange in and out of the bloodstream is at a capillary, except for these white blood cells. They can enter tissues from any blood vessel. So generally, they don't get in or out at blood vessels, especially capillaries. Usually we keep our reds in at our capillaries, but the whites have the option to go in or out wherever they want. So diapedesis is that ability to move through a blood vessel in order to get in and out wherever they want to. So they sense damage, they sense a problem that they need to be involved with and defend against, and they start to head towards that. Once they find it, they hop out of that bloodstream wherever they're at because they ain't got time to fool around, right? Chemotaxis. Positive chemotaxis is a better term. Chemotaxis. Taxi means to move. Chemo, chemical movement, positive, towards. These guys are attracted by chemicals that other cells make, other damaged cells or pathogens make, or other white blood cells make. And they're like bloodhounds, and this is their trail. So they follow these chemicals to the infection site, to the damage site, and that's how they know where to start fighting. They smell the damage. They smell their buddies working and know that they're working hard and they need some help, right? They smell the bad guy. And so this is how they're attracted to this area, and then they start whooping some butt, right? Now, I'm not going to hammer this on the test. You can learn it if you would like, but I'm not going to hammer this. White blood cells are divided into 2 groups, granulocytes and agranulocytes. And it all depends on if they have grains inside or not, right? Well, there's 3 granulocytes and there's 2 agranulocytes. Now, if you really want to learn this, you can. Again, I'm not going to focus on the test on this. If you're a fill, then you're a grainy, right? You're a grainy fill. If you're a cyte, then you're not grainy, right? So that's a way to remember it. Neutrophil, eosinophil, basophil. These are granulocytes. They contain grains inside. They contain vesicles that are going to contain chemicals for their combat, for their defense. The agranulocytes do not contain these chemicals. They're going to do something else. Monocytes are our big eaters. Lymphocytes, these guys are going to be our specific defenses. So let's talk about them real quick. Neutrophils, these are the most numerous, and again, we don't have as many whites in the blood as we have everything else, but these neutrophils are the most numerous white blood cell in the bloodstream. And so these guys are the first line of inner defense. First line of inner defense because their numbers rise dramatically during bacterial infections, especially chronic. So if you go get your blood panel read and you got a high PMN number, a high Newt number, then that means that you have a long-term infection. Okay. Neutrophils are the first to appear. They love to eat bacteria. These are frontline infantry. These are the grunts. They know they're going to die, but they jump out of that helicopter and race into battle because they know it's the right thing to do. So these guys are very chivalrous. They're also called PMNs. You don't need to know that because they have many shaped nuclei, polymorphonuclear, but I'm not too concerned with that. These guys are, and what I do want you to know about them, this is our primary group. This is the main group of macrophages. So these are the main small eaters that we're going to talk about, the macrophages. And so these guys can enter into tissues, and there they can start to eat these bacteria. They're going to eat bacteria until they die, usually a couple dozen. They don't eat very many, but we've got a bunch of these guys. So a couple dozen bacteria, and then they start to die. And if they don't exit that area, then they can start to become encapsulated. And that can create pus. So pus, the whiteness in pus many times is from dead white blood cells, neutrophils. And so we can see that neutrophils are very important to defeat that bacteria. Now, one thing that I want you to write down is the term respiratory burst. Okay, respiratory burst. This is a method by which this neutrophil engulfs a bad guy and increases its metabolism, increases the neutrophil's metabolism to the point where the bad guy can't take it, and then he dies. So this is one of the methods, the respiratory burst, that a neutrophil will use to destroy its target. There's a neutrophil. You can see how big the whites are compared to the reds. And again, neutrophils are the most numerous. That's all that I want you to know. You don't have to know percentages. Eosinophils, these are at a minor percent. You don't have to know specific 1 to 4. Just know minor percent of the whites in the blood. Eosinophils, there's a stain called eosin, and that is a red stain. And so these guys stain with that red stain, and they look reddish. And so that's the reason they got their names. These guys have two main functions. The first down here, these help fight multicellular parasites. We like to call those worms usually. Most of our multicellulars are worms. And so they help to destroy these multicellular parasites. So tapeworms, if your dog has heartworms or have intestinal worms, then these are the things that help to fight that. This is the only thing that I know of that helps to fight multicellulars. Most of what we have to fight off is single-celled, bacterial, virus, fungal spore type things, okay? The other thing, it helps to control allergies. So eosinophils are going to increase during an allergic reaction, and that's going to help to control the inflammation, help to control the allergy from that antigen and antibody binding together. It's an overreaction. That's what an allergy is an overreaction of the immune system. So these guys are kind of suppressors. They're kind of down regulators of that. Here we can kind of see that reddish stain in that cell. Basophils, again, minor percentage. I want you to know that they create inflammation. These guys cause inflammation. Now, they cause inflammation by releasing two chemicals, histamine and heparin. And you need to know what these two chemicals are doing. Baso, you don't need to know it. It's a blue dye, so these guys kind of stain blue. Who cares about that? We want to know main functions. Here, histamine and heparin. Histamine, I want you to know, causes vasodilation, causes an increase in the diameter of the blood vessel. It opens the blood vessel. So histamine is a vasodilator. This is what causes those allergic symptoms, right? There's allergies. You get the itchy red eyes, right? You get an itchy red nose, itchy red nasal membranes, right? That's what histamine is doing, is it's increasing that blood vessel diameter, and it's creating those problems. And again, this is due to an overreaction, really, of that immune system. Heparin, this is an anticoagulant. It inhibits blood clotting. If we're going to open up blood vessels, then we want to not have clots. So we want to inhibit blood clotting. So heparin is used to limit that. Now let's talk about our two agranulocytes. Lymphocytes are the second most abundant, okay? So here's how I want you to remember this for these white blood cells. Number one, neutrophils. They're the most abundant. Number two, lymphocytes, second most abundant. All the other three are just considered minor percentages, okay? Just minor percentages. So these lymphocytes, these guys usually hang out in other organs. These are our special cells. These are the special defenders. These guys, the only one, there's three categories: T cells, B cells, and NK cells. NKs attack abnormal and infected cells. So these guys are super important. Abnormal, cancerous cells, infected, bacterial or viral infected, COVID infected, right? So infecting those cells What's going to kill them are called natural killer cells, NK cells. But these other two, give yourself a note, the T's and the B's, these give us what's called our specific defenses. Our specific defenses by T and B cells are our true immunity. Without these, we can't really fight off anything and destroy it. And so these are our two most important cells for our immune system. T cells are going to be cell-to-cell combat, cell-mediated immunity. B cells, these guys are going to make antibodies. So T cells physically enter a tissue and beat its butt. They do the same thing that NK cells do. They attack these abnormal and infected cells. But these B cells, they hang out in the mastermind room, in the think tank, and they're just making antibodies. They're coming up with these little heat-seeking missiles that always finds that antigen, that foreign antigen, and if it binds to it, we've already seen we're going to get agglutination, we're going to get chopping, we're going to get destruction, kind of like with that bad blood transfusion. So here we can see those lymphocytes. Last group are monocytes. Monocytes, these are also called macrophages. So I want you to know that monocytes are the same thing as a macrophage. So these are our big eaters. Again, the neutrophils were our main macrophage, our main little eater, and these monocytes are the big macrophages. Now here's the key. If it's in a tissue, it's called a macrophage. If it's in the blood, it's called monocyte. They are the same dang thing though, okay? Monocytes and macrophages, it's just where you at. In class, I call you a student. At home, you're called mama. At work, you're called a piss on, right? Just kidding. But you got a different name depending on where you're at. So monocyte in the bloodstream, macrophage in the tissue. These are the big eaters. Big eaters have two main jobs. They are policemen and they're janitors. So these guys are going to eat bad guys. They're going to eat bacteria or viruses. But they're also going to help clean up. Because after some of these immune battles we've got a real littered battlefield, and we don't need to have another infection set in because of that. So they're going to help eat debris too, kind of like a janitor sweeping up, keeping an area clean so that we don't have rats and we don't have all kinds of evil kind of diseases showing up. So these guys, super important for eating bad guys and eating cell debris to kind of clean up an area. Now, I want you to be familiar with these two words, leukopenia and leukocytosis. We're only about 10 slides from being finished. We got to focus on platelets and blood clotting real quick, and that shouldn't take very long. But here to wrap up our whites, we're going to look at leukopenia and leukocytosis. If you've never heard this suffix penia, it always means low, and cytosis many times means high. So leukopenia means low white blood cell count. Okay, now we may be more prone to infection. Leukocytosis means elevated white blood cell count, and this could be a bad thing. This could actually be due to infection or stress, but this could be if it's too highly elevated. Now we could start autoimmune problems where we start to attack good tissue and start to destroy it, even though we shouldn't. Now, again, I don't care if you look at this, but the differential count, this is something, for example, that you might see if you get your blood panel read. Here is approximate numbers. We're looking at in the millions, 4.2 to 5.5 million for reds. And here we're looking at less than 10,000 practically for the whites. So the point is that there's not a lot of whites compared to reds. Majority neutros, second most lymphos, the rest are minors. That's the way I want you to think about them as far as percentages. Now let's hit these platelets and let's get them over with so we can get finished with today's lecture. Platelets, don't forget, are involved with clotting. Now, if we look at numbers again, compared to the 10,000 number that we just had for whites, we're looking at hundreds of thousands. So these are more abundant than the whites in the blood, but again, the reds are in the millions, so these are nowhere close to the millions, okay? So platelets, again, these are cell fragments. They are not actual cells because they're missing their nuclei, so we can't really call them cells. Important for clotting. And the spleen is where we tend to keep most of these. They don't live very long. They circulate for 8 to 10 days. We're going to break them apart and rebuild them, regenerate them. Thrombocyte. You haven't heard that word yet, but I want you to connect that word. So right here we see thrombocytopenia. Drop the openia and put an E, thrombocyte. That's another term for a platelet, especially in another animal. That's a good term for them, is a thrombocyte. Okay, so thrombocytopenia would mean low platelet count. Thrombocytosis would mean high platelet count, for example. Okay, so low platelet, you're not going to be able to clot. High platelet count, you're throwing clots, and you don't need to do that. Okay. Again, these are cell fragments. They've been destroyed. They've been emptied of a lot of their parts, and now it's really just kind of like a membrane with clotting stuff inside. And we call that clotting stuff a clotting factor. Okay. Now, to start wrapping this up, hemostasis. Hemostasis is the process of blood clotting, and I'm going to simplify this. There's three stages. There's the vascular spasm, the platelet plug, and the coagulation phase. So this is really not that difficult. There's an image in your textbook. Here, I'm going to flip to it real quick. So here you can kind of see mine, again, has been screwed up for some reason, switching it to a PDF, but it's showing us the three different stages. Now, as we go back, let's go back through these and let's talk about them. Oh, sorry. Okay, so our first stage is the vascular spasm. The vascular spasm is after we cut the vessel, we're going to have a spasm in that vessel. So after we cut the vessel, it's going to constrict. Now what this is trying to do, it's trying to cut off circulation, limits the blood leak the vessel. So now this is what most people think is a clot. It's simply just the vascular spasm. It doesn't take long. It's a very quick, immediate reaction, and it lasts until the true clot is kind of put in place. So we're going to constrict that vessel, and constricting it means now we got less blood flow. Then we're going to start to plug the hole with platelets. So we actually physically come in and we start to plug that hole. Let me go to that picture. You can see right here, we cut the vessel. Now we're plugging it with platelets, and this is what's called the platelet plug. Anything that's exposed, any part of that vessel that's exposed, these platelets start to stick to it. And then they start to build a bridge across to stick to themselves, and so now they've filled in that gap. But this is not a true clot yet. Okay, true clot is coagulation. All that we're going to do now is add one more thing, fibrin. Okay, so this is the most important and complex component. It's very complicated, but I'm going to simplify it by saying we're just going to add fibrin. To what we've already got, we add big logs. We add this fibrin, this network, and now we're going to start to trap other things and start to create that big beaver dam, and now be able to hold that blood back and keep that blood in that vessel longer. Now this coagulation, this is not permanent. We don't need this clot forever. We want to regrow the vessel back together. So we're going to try to constrict and pull that clot back together. Okay, again, clotting factors, any substance used in coagulation. Calcium is a clotting factor, vitamin K is a clotting factor as well. As I mentioned, that clot is not meant to stay there. We're trying to eliminate the clot to get back to the normal vessel, right? The perfect vessel wall. So when we try to eliminate that clot, we're going to retract it, and then we're going to undergo fibrinolysis. We're going to take away some of that fibrin. So the clot retraction, we're kind of squeezing in that area a little bit with that smooth muscle, and we're trying to shake off any extra stuff, anything that's not needed, anything that's out of that vessel. Well, we want to contract and squeeze it and make that clot smaller. Okay, so we're trying to make this clot smaller and more efficient. And then eventually we're going to take away the fibrin. And so we're going to take away that fibrin, and now the vessel wall is going to start to grow back to itself more completely. Okay, so clot elimination, it's not a bad thing. Clot retraction, right? This is kind of the final step to getting rid of the clot and making sure that the vessel grows back together properly. Last thing on our list today is to quickly look at hemophilia. Hemophilia, genetic bleeding disorder. So the problem is that you do not have proper clotting factors. So hemophilia, you have a deficiency in certain clotting factors, and as a result, you end up bleeding more often. You get a cut and you bleed more. So this is an X-linked recessive gene. What that means is it's carried on the X chromosome. Females have to have two X's to be a female, and males only have one. They have an X and a Y. And so the problem is that if you only have one X, then you've got a better chance of showing that disorder. And so because males only have one X, they tend to exhibit this disease more often. Males tend to be more hemophiliacs, and they tend to be more colorblind than females. Females, on the other hand, they need two X's to be recessive. And so they tend to not have that. They tend to only be carriers. So mama doesn't even know that she's a carrier until she has a baby, and that baby, that man, that little baby boy has hemophilia, and they don't realize it. So at that point, we have finished this blood chapter. Okay, that is it for chapter 18. That is it for our first test, right? So get on those study questions. If you haven't been studying much yet, you are so far behind. You got to get the hot foot going, girl. So make sure that you are doing your best to stay up with this material. You're taking good notes. You're using your notes to answer your study questions. And there you're studying both your notes and your study questions to get prepared for the lecture tests. Okay, lecture test is almost entirely multiple choice and maybe some matching or true false. And so be prepared for anything there that you can just kind of put a simple answer to, right? All right. So get studying. If you have any questions, feel free to contact me. And I hope that you have a wonderful rest of your day. Thanks a lot, y'all.